For the morning. Let me just make sure though that I've got my, not quite yet. There we go. Okay, great. Let's get started. I'm John Novembre. I'm a population geneticist at the University of Chicago. And today I'm going to be talking about population structure and some of the tools that we use to study it. So population structure is the existence of some form of non-random mating that is due to subpopulations existing within the population, some form of restricted dispersal. So these are some of the classic examples of population structure that have been well studied, such as the island model. This is a symmetric island model where you imagine a number of subpopulations, each exchanging migrants with the other one. So most matings would tend to be local, and then migrants get exchanged to neighbors, equally to all others. Then there are what are called stepping stone models, such as the one-dimensional stepping stone model. The name comes from Japanese gardens, introduced by Moto Kimura. So these are each a population in which migrants would move to a neighboring population in that abstraction. You can have two-dimensional stepping stone models, and models where you have an ancestral population that then diverges into two subpopulations. This is sometimes called the pants model because in certain ways of drawing it, it can look like a pair of pants. Then you have admixture, where two populations contribute individuals to an offspring population. Okay? And actually, you can put a set of these together to make a larger tree structure, right? And these admixture edges to make a graph. Okay? Okay, so that's just some basic forms, but of course reality is more complicated, and sometimes it doesn't fit into one of these canonical forms. Some of the major tools that are used for studying population structure include admixture proportion inference. So this is something I'll talk about more, but each of these vertical bars represents an individual, and the colors represent their proportion of ancestry from various ancestral populations. Ordination methods, such as PCA, where individuals are plotted along a small number of axes to represent their similarity genetically. And tree-based models, like TreeMix here, where a tree is inferred for the data, possibly with admixture edges to reflect admixture events that have occurred above and beyond the divergence relationships. Population structure is interesting to study broadly in evolutionary biology, ecology, because you can't understand how populations are going to evolve unless you understand what units exist and how they're exchanging migrants with one another. You can't manage a population in terms of its ecology if you don't understand how individuals are moving between populations and what units exist. And then for many of us here who work closer to human genetics, it's important for understanding in terms of its role in affecting genome-wide association studies. So genome-wide association studies, we've only been talked about briefly here, but the idea is that you're modeling some phenotype in a linear model type framework. We have some mean, some observed covariates, and what you're interested in is testing whether any given SNP along the genome has an impact on the phenotype. So it has the look of doing a regression where you're interested in this regression coefficient, whether it's non-zero, and this is the negative log 10 p-value that's plotted across the genome for that test. The problem is that if the underlying phenotype has some factors that contribute to its variation that are driven by population membership, so for instance environmental factors that vary across populations, or even a polygenic background, some genetics that vary by population that are not the SNP that you're testing, then if the SNP that you're testing varies across populations, it can end up standing in for population membership, okay? So that you will see a significant SNP. If you don't include population membership as a variable when you run your genome-wide association study, the SNP will stand in for population membership, and you'll get a false association, okay? So what is done to correct this is to put in extra variables that represent population membership, either in the form of ancestry coefficients, principal component values, or random effects that have a covariant structure that is meant to model the population structure in the data. Okay? So in short, just in terms of motivation, right? If we don't understand this population membership, we'll get false positives in genome-wide association studies. Yeah?
So how does it work if that SNP is actually really effective? Like, it's actually the causal SNP,
Yeah.
so it also does have the correlation with the population.
Yes. Yeah, so that's you have an issue with power there, right? So if the SNP correlates with population membership and it really has an effect, it'll be difficult to untangle what's population membership versus what's due to that SNP. And usually the corrections for population membership do weaken your power to detect those. That's a case where finding some way to randomize the individuals across the environments would be ideal. It's a limitation of observational studies, but if you were in plants or something like that, you could do some sort of work to randomize the genotypes. Okay. Okay, so today what I want to talk about is an introduction to fixation indices. These are a tool used to measure population structure, and I'm going to spend maybe a good amount of time on this because it's not a topic that's that you see taught very often. I thought it's a good venue for it. Then I'll talk about structure admixture models in PCA and try to connect them with a view of seeing them both as factor analyses, and that'll be a unifying framework. Then there's a lot that's known about PCA and that I've worked on, so we'll talk about some noteworthy behaviors of PCA. I'll mention this idea of effective migration surfaces very briefly and then conclude. Okay. So let's just think about a single SNP, okay, and which we can summarize with genotype counts. So an AG SNP where we have 64 individuals who have the AA genotype, 32 have the AG, 4 who have the GG in one, say, population. And then in the other case, we've got 72, 16, and 12, okay? So already from this data, you can detect population structure, okay? If you know what you're looking for. Can anybody spot it? What's that?
Second population is more homozygous.
Okay, yeah, the second population is more homozygous cases, and you think that might be a signature of population structure? Of a small population, okay. Yeah, so some small populations might have more heterozygous or homozygotes, but it could be small population and be completely random mating within, and that might lead to excess homozygosity. So it's a true observation. These do have like an enrichment of homozygotes. Yeah. Yeah, and they're out of Hardy-Weinberg, and yeah, what were we going to say in the back there? The same, yeah. So if you're quick with math, you can catch that this is the allele frequency here for A is 0.8, and it's actually 0.8 in both cases. But in this case, if we take 0.8 squared, and there are 100 individuals here, 0.8 squared, 64 times 100, we get 64. So this is then 2pq would be 32. Q squared, the 0.2 squared would be 4. So this one's exactly in Hardy-Weinberg equilibrium, which is to say that the genotypes are paired exactly as you'd expect if you independently drew alleles for each allele in the genotype. Okay. Here we see relative to these values an excess of homozygotes. You're right there. Okay, and that can be a signature of population structure because it may be that there's a subpopulation that has lots of A's, and so lots of AA homozygotes, a subpopulation with lots of G's, and so lots of GG homozygotes. And the two subpopulations, when looked at together, would not be producing the patterns that you'd expect from independence. You'd see this excess of homozygotes. Okay, we're gonna rehearse this and hopefully understand it more formally. Okay, so. Yeah, this is what's great with being a mathematical quantity. We can introduce some notation and think about this carefully. So if we put an indicator variable, just 1, 0, for whether the maternal allele carries an A. So let's make this the maternal and this the paternal. And then another indicator for whether the paternal allele carries an A. Okay, so 0, 0 would be the genotype GG, 0, 1 GA, 1, 0 AG, 1, 1 AA. Okay. Then we can think about each of the variables on their own, indicator variables. Their expectation will be equal to the probability of seeing an A allele, which here we're just going to denote as the frequency P, so P is the frequency of the A allele, and it'll have a variance, okay? But what's interesting here is the correlation between these two variables, okay? This is sometimes called the correlation of uniting gametes because we're thinking about the maternal allele and the paternal allele had to come from one gamete uniting with another, right? Okay, so this correlation we're going to define as a variable f, okay? And so in large populations with random mating, no selection, no mutation, should the value of f be 0, positive, or negative? And then if a population has some structure where there's some subpopulations within it, should it be 0, positive, or negative? Okay. So answer to the first question is 0. Okay. That's the, in essence, ahead of realizing this term f, but what Hardy-Weinberg found. Okay. That with a basic set of assumptions that the alleles within genotypes should be as if there is no correlation between them. They're independently sampled, like indicator variables. Okay. But if we have structure, as we were just saying, you might have the big A alleles paired with big A alleles in excess, right? And G alleles paired with G alleles in excess. Think about it this way, that in a situation where say there's A is common on this side of the room, so we should have lots of AA homozygotes even if we're randomly mating. G is common on this side of the room, right? If you tell me that the first allele, a maternal allele, is an A, I automatically know, oh, you're probably from this side of the room, and so your paternal allele is probably an A, right? And so there's some shared, some information there. There's some correlation between those two indicator variables that's introduced by the clustering of alleles geographically. Okay, so when there's population structure, F will tend to be positive. Okay, so then this correlation statistic F actually turns out to have some nice uses. One of them is what's called its role in what's called the generalized Hardy-Weinberg equations. So if we have some correlation between the united gametes, so this F is not equal to 0, we can write out the probability of seeing an AA genotype, a big A little a, or a little a. So I've switched from AG to A little a, in terms of these equations here. When F is equal to 0, these are just the classic Hardy-Weinberg, which are also what you would get if you think about sampling these indicator variables together. So you would have, with F equal to 0, you'd have P squared is the only term here, right? So that's a probability of seeing an A and an A, right? Then with F equals to 0, this would be 2P 1 minus P, which are the two different ways of sampling a big A and a little a, right? And then little a is this kind of symmetric to this case. And so when you see when there is this, it's probably easiest to focus here on the heterozygotes. When F is positive, there's a correlation between these alleles, then the heterozygosity, how many heterozygotes we expect to see proportionally, is going to be less than what we'd see at random. Okay? And to compensate that, you'd have an excess of homozygotes. Okay? We'll see that more in just a second with some graphs. But before I show the graphs, what this also highlights is that we can estimate F, this correlation, in terms of taking the observed proportion of heterozygotes and subtracting it from an expected number of heterozygotes based and using an estimate of the allele frequency from the population. Okay? So this is sometimes denoted as the expected heterozygosity minus the observed heterozygosity over the expected heterozygosity. Okay. Great. Okay. Good. I've said all that. Okay, so what does this look like in practice in some human genetic data? So this is just, oh, we got a little problem with there, I'll just put them both up. This is from the Human Genome Diversity Panel, taking real SNP data and plotting as each SNP as a set of 3 dots. Okay, so we will compute the frequency of one of the two alleles, the minor allele, actually the allele that's at lower frequency, and we'll call that the big A allele. And we will show here for each SNP at its frequency what proportion of big A genotypes there are, big A genotypes, and then little a. Okay? The thick lines show what we'd expect if there was complete independence amongst the alleles. Okay? This is p squared, in a nice quadratic form. P squared 2p 1 minus p. P squared 1 minus p squared 2p 1 minus p. Okay? And actually you see that across thousands of SNPs here, the data fit quite well in terms of the expectations lining up really nicely with what you'd expect in theory. And if we calculated F for this data, it would be about 0.003. Okay? So very small. So real human data actually fit quite well to the classic Hardy-Weinberg proportions and show no correlation of alleles within regions at the scale of Europe. Okay?
This is across all the SNPs on chromosome 15.
Yes. Okay. Then if we look instead at global populations, so now we zoom out, we see that there's actually the data tend to sit below the line for the heterozygotes and above the line for the homozygotes. So we're seeing that deficiency of heterozygotes that's typical of the alleles being correlated, right? So having a big A for one parent makes you more likely to have a big A for the other, and it gets you this excess homozygosity. All right? And the F that we compute here is about 10%. Okay? So this deficiency of heterozygotes is about 10% of what we see here. All right? This is a real absolute measure of population structure. It tells us, in some sense, humans are only globally are only 10% away from what you'd expect if we were just completely randomly mating. Okay? That's really striking. It must indicate that there hasn't been a lot of time for allele frequencies to change since we all began to move out across the globe. Yeah.
Is there a reason for the heterozygous cases to have higher variance there?
Higher variance here.
Yeah.
It's an interesting question. The variance is going to go with p times 1 minus p, but I'm not sure exactly. Yeah, and we can look at that. Okay, so great. Okay, so this is a really nice absolute measure of population structure. It has a relationship to thinking about how alleles pair into genotypes, a very fundamental aspect of population genetic data. Oh, in terms of learning some terms used in the field, the Volland defect is the name for this deficiency of heterozygotes that you see when you have population subdivision. Okay.
If the variance is p times 1 minus p is maximized at half, right?
That's right. Yeah. So seeing this spread, yeah, but I'm not sure why. Like, the variance on the p squared, I'm not sure if it should be relative to that off the top of my head. Okay. So. Okay, so now if we return to these two cases, p is 0.8 for both of them. If we compute f in this case, it's 0. If we compute f in this case, it's 0.5. Okay. So even with just simple genotype count data, you can already detect population structure. All right. Now, what if we have this is also with no population labels. Okay, what I just showed you. But what if we have subpopulation labels? Okay. So we have, imagine, say, one population. A that has this set, another population B that has this set. And let's say this has a frequency of 0.8, this one a frequency of 0.2. Both of them have an F of 0, like within these two, we'll call them subpopulations. And we look at the total population, so we put these two together and then sort of sample 100 individuals, we'd have 34, 32, and 34. The allele frequency now is 0.5, and F in the total population is 0.36. So we can see we compute this F at different levels of hierarchy. We compute it within subpopulations. We can compute it at the total populations. And we can see that we can have no correlation of alleles within populations, but it can arise once we go up to that higher level. Okay? This underlies a larger framework of thinking about these correlations in which we decompose the value for the total population into two parts, okay? And you can actually do more if you have different, more complicated hierarchies, but the kind of classic decomposition is to break it into the correlation that's due to inbreeding within subpopulations. So if we had seen F not equal to 0 here, okay, then we would maybe guess that there is something else contributing to the excess of homozygosity, which this correlation, and that could be inbreeding. So if individuals are mating with their cousins, that will lead to an excess of homozygotes that doesn't have to do with population subdivision. Okay, so so we can break up the overall F, which is in this more general framework called FIT, into a component that's due to the inbreeding within subpopulations, which here is 0, and then the contributions that's due to the existence of subpopulations, FST. Okay, and so in this scenario, all we have is a contribution from FST. This FIS is 0, so the F overall is really driven by the FST. Okay, and in practice, like in a lot of, say, like human genetic data, FIS is really quite small. I already made the point that like F due to population subdivision is kind of small, like 10%, but you know, the FIS is going to be really small. All right. Okay, so great. So in terms of applying these, F, this what's called the fixation index most usually, provides a tool for detecting general deviations from random mating and Hardy-Weinberg conditions even without subpopulation labels. Okay, that can be due to population subdivision. It could be due to inbreeding. It can actually be due to other forces as well, but it at least tells you that you're away from Hardy-Weinberg. Okay, FST, if you have population sublabels, can tell you specifically about whether there are a departure from independence that's due to population structure. Yeah.
Is F always positive, or it could be negative?
It can be negative, although it's tricky to come up with population genetic models that, like biological, will induce a negative F. Okay. Yeah.
Does it make sense to compute this F for like certain frequency spectrum?
Yeah. So.
Specific history or biological?
Yeah. So in basic models, it depends on the exact kind of population model you're interested in investigating. If you have just simple population structure, like these random mating populations, subpopulations are part of a total, you'll get the same F regardless of what allele frequency you use. But in practice, maybe there's some non-equilibrium history where things have been taking place at different time scales, and then some deviations might be more strong in the common alleles in the rare, or vice versa. So there are interesting things that could be done with that. Yeah. Okay, so this F is a really fundamental quality in population genetics. I introduced it in the context of population structure, but it's much more broadly interesting. And I kind of just leave this to you as like if you want to follow these paths later. Introduces correlation, how I did it here, but it's related to inbreeding coefficients and pedigrees. If you've ever calculated those, you've seen F come up in that context. Fixation indices, as I just called them, is how Sewell Wright introduced them in the terminology. Working independently, Gustave Mallico came up with probabilities of identity by descent, which turn out to be the same thing. And there's a tight connection to pairwise coalescent times, because how recent the pairwise coalescent is relative to the rate of mutation determines how correlated two alleles that are sampled will be. And if you make like an ANOVA-like analysis of population structure where you have labels for each population, then F, and specifically FST, will predict the proportion of variance observed between subpopulations. Okay, so this one number really unites a lot of different ways of conceptualizing population genetics, and has a lot of interesting history and theory behind it. Okay. In practice, though, for many people's purposes, is it greater than 0? And what's the number to say, like, oh, that's a lot of population structure? You have an FST of 0.5 versus, oh, that's not much. You've got an FST of 0.003. Okay. And the estimation of the value has some subtleties when sample sizes are small or when rare variants are present. Okay, and I'm not going to get into those issues. They're more kind of in the weeds statistical issues. But there's a nice review in Nature Review Genetics by Holsinger and Weir, and then a recent paper about the issues with rare variants by Batia et al. Okay, yeah.
So what do you think about the use of FST as a distance measure between individuals or between an individual and a population or between populations? There's a fundamental issue that sort of diverse populations appear close to everything, right? If you use it as a genetic distance.
Yeah, yeah.
Do you have a scientific remedy for that, or should it just not be used?
Yeah, one has to be careful. Yeah, I mean, at short divergence times, it can be reflective of the genetic drift, and how much time has passed in a very sensible way. You can, from a population genetic model, derive how FST relates to a divergence time. But but as you get to larger divergence times or have migration, it gets more complicated. Yeah. So I don't tend to use it in those forms, and won't talk about it in that way. But I think it is just a good representation of the absolute level of differentiation. Yeah. Okay. So now we're going to talk about two of the most commonly used approaches. So I'm not going to talk about tree-based methods, and that actually connects to Sharhan's question. We're going to talk about individual-based approaches for looking at population structure, because these are two very commonly used ones, and admixture proportion inference and these ordination methods. Okay. So both of these methods take as the basic input a matrix where rows are individuals and columns are SNPs. And I like to think of it, conceptualized as a matrix of 0, 1s, and 2s. We're going to make this what's called an additive coding, where we represent the genotypes by counting a reference allele, and so that the heterozygote has a 1, the homozygote for the A has 2. Okay, and this will be an n by p matrix, or n individuals, p SNPs. So now p is not allele frequency; it's the number of SNPs. Okay, kind of keep with the more general notation used in many areas. Okay, so with admixture proportion inference, the idea is that you create a model where there are k ancestral populations. So here k would be equal to 4. And for each SNP and each population, there's an allele frequency. Okay? And then your study individuals are modeled as having a vector of proportions of how much of their ancestry is from each of these source populations. So this individual would be, say, maybe 40% from this and 60% from this. This one, 90% from this one, 20% from this one. And so the goal is this is a stacked bar chart representing this vector of admixture proportions, and the goal is to estimate those admixture proportions. Okay? It's a vector oftentimes labeled Q of length k per individual. Okay? And the underlying model, I'll say a little bit more in a second, models genotypes, with allele frequencies in each of the populations, ancestry proportions, and it assumes, basically, the only assumption in this model is that the alleles are inherited independently. Okay? And it's, once you know the allelic proportions, it's like one allele, each allele draws, based on the ancestry vector, a source population, and then you draw an allele from that population. Okay, and then the next allele is doing that independently. So in a way, if you have this type of data where there's excess correlation of alleles within genotypes, it's going to try to kind of force the individuals, like say this situation we talked about before with lots of A's over here, lots of G's over here. If we model the data as individuals over here have their ancestry from one population, individuals from over here have their ancestry from another population, then the kind of residual correlations will go away. Like we'll be able to, it'll look like two random mating populations. All right. So the original approach for doing this model fitting was using MCMC, a Bayesian approach, in the software Structure, which has had over 10,000 citations, probably, 15,000 by now. This is already an old slide. And then once large-scale SNP genotyping data became available, these approaches became too slow, and so alternative approaches using an EM algorithm with the maximum likelihood formulation were developed by Hua Tang's lab at Stanford. Then here at UCLA, admixture was developed by David Alexander and Ken Lang, and I participated. Then and then the structure authors have produced recently a variational Bayes approach called fast structure. Okay, and there are a bunch of other actually variant approaches, but these are some of the main ones. And I want to learn more about this. I've written 2 reviews about this recently. So one, a news and views really on the fast structure paper, and then the other was for a special issue celebrating some of the most cited papers in genetics. And so this has some nice history about the structure paper. So one way to write the model here is to have the genotypes, these 0, 1, 2s, be conceived as being binomial draws of sample size 2 with a particular underlying mean or success probability. Okay, that success probability is given by a sum over populations of the allele frequency for the jth allele in the kth, or at the jth locus in the kth population. Okay, so the allele frequencies across populations weighted by how much that particular individual I has ancestry from that population K. Okay, and yeah.
Q is constant for all. That Q is constant for all, for that individual, for
all loci. Yeah, yeah, yeah. Okay, yeah.
So this model assumes that the mother and the father comes from independent ancestry because it's binomial 2 RIJ, right?
Yeah.
So that means if your mother is African American, then your father is independently drawn from the same distribution. There's no correlation between whether your mother is African American.
They're drawn independently, but from the same ancestry proportion vector. So if you're, so it's, but yes, I mean essentially yes. But the individual has say an ancestry proportion vector.
I see.
So
Okay.
they would have like 90% African ancestry, then that would get sampled twice.
Oh,
Yeah.
okay.
Okay.
Kind of assumes that the mom and the dad have the same underlying proportions as the new person that's being created.
That's right. Yeah. Yeah. Okay. Then okay. Now, if you look at this, you can see that it is. It can be written as a matrix multiplication, where we have a matrix Q. If we put these in a matrix Q, put these in a matrix P, okay, then we can have a Q matrix that's n by k, a P matrix at k by p, and have the expectation of the genotype be this product of 2 matrices. Okay. Yeah.
Why not make it Bernoulli and do it separately for the mother and father? Isn't that the most standard?
So maybe I'm pushing things a little hard here. So in the original structure model, there's actually a latent state for each variable that is its ancestry. It is sampled independently from these Qs, and this is sort of marginalizing out that latent state. But you can.
In the next paper, I think you have separate variables for the mother and father, like an additional index.
Yeah, so you can do that. Yeah, sorry, like sorry, did you say in the Yang paper?
Thanks work.
Oh, in Watang's paper. Okay.
The third index for the mother versus father.
Okay. Yeah, so there are some variants that can do that. Yeah. What I'm pushing at is that at least with like a basic a version of this model is going to be can be understood as factor analysis. Okay. So this is just, but it's true that like different variants of these models can have slightly different flavors. Okay. So factor analysis is the idea of modeling the mean of multivariate data as a product of 2 matrices, where you have a set of factors that are a set of vectors that are in the feature space, so they're length p, and then a set of loadings per individual, and so with this structure can be seen as a factor analysis. But we have constraints here that in here, these Q are a vector of proportions. So the rows of this sum to 1. And then these factors, being allele frequencies, are also rows that sum to 1. Okay, so yeah, so it's a kind of constrained factor analysis. All right. Now let's turn to principal components analysis. The classic way that it's introduced is to think about multivariate data, say here 2 dimensions, that has some covariance structure. So here where there's more, there's a positive covariance and between the two, and you see this long axis where there's more variation than along the original observed feature dimensions. And so the first principal component will run through here. The second principal component is chosen to be orthogonal to the first and capturing those variants. So it's kind of, it's automatically just this axis in a 2-dimensional case, but in higher dimensions it can be more interesting, and we'll choose the next axis that has the most variance. So, and then the data are then rotated onto these new axes, PC1 and PC2, and this is a representation of the data that more directly shows on the PC1 axis the most variance, the PC2 lower dimensions. And the idea is that like I could throw away PC2 and still be capturing a lot of the variance on PC1. Okay, I can use it for some kind of compression or more sparse representations. In the context of SNP data, what it looks like is we have our individual by SNP matrix, and then for each individual, we're going to take this linear combination, get it down to a particular principal component value, and oftentimes what people do is just do 2 or 3 or 4 and plot the PCs one against the other. Okay, so this is a data matrix from simulated data where there are 2 source populations and a set of admixed individuals, and so it nicely puts the admixed individuals in between the 2 sources. Okay, now how this is done in practice is to take the original matrix, center the columns, and scale the variance to 1. Then take the this outer products matrix, which is a covariance matrix, and diagonalize it. And then the columns of this will give you the principal component, the projections of those individuals onto the principal component vectors. This will be a diagonal matrix with eigenvalues that tell you about the proportion of variance explained by each of those principal component vectors. In practice, this is computationally expensive, so what's typically done is to do this via singular value decomposition. So if you do an SVD, you get a matrix that has its columns being the individual projections onto the PCs, which is what you'd use for these individual level plots that are used in population genetics very often. Okay. And you get the eigenvalues in this S matrix. You get the vectors that define the linear combination of SNPs, the principal components in this vector. Okay. This can also be seen as a factor model. So if you make a probabilistic formulation for PCA where you're having the normalized genotypes being modeled as in a factor form where you've got loadings and factors, but they're constrained to be orthogonal and orthonormal, then the MLE solution for this will be the terms that you see in the singular value decomposition. So the U matrix, which has the vectors of 1 per individual of length k, and then the SVT kind of combination matrix, which has the vectors of length p. Okay, so yeah.
For each pair ij, these are independent and equally distributed, right?
Yeah.
So if you look at all the data, the variance matrix, it's the factor structure plus the white noise.
Yes, yeah, in this form, yeah, okay. And this Engelhardt and Stevens paper has a nice discussion of this like interpretation of PCA as a factor model and admixture as a factor model, and then once you have that, you can start playing tricks like, well, what if we make like different assumptions about maybe the sparsity of the elements of these matrices and so on, okay. This is an example of PCA being applied to European data that I was involved in, where we were able to look at a large number of individuals from across Europe, and what's shown here is the PCA on their genotype data matrix. Each 2-letter code is a single individual. The median position of each individual is shown here with a large dot. The median position of all individuals who have the same code are shown with the large dots, okay? These codes correspond typically to country level ancestry labels with a legend given here, okay? The ancestry labels are based on individuals' grandparental ancestry, and we're only showing individuals where all of their grandparental ancestry points to a single location, okay? So we can give one label instead of like hybrid labels. And what you see is this remarkable similarity in the genetics to geography, okay? And in terms of the proportion of variance explained by these PCs, it's very, very low, 0.3% for PC1, 0.15% for PC2. Yeah?
I was wondering, can you ever go backwards to kind of figure out which SNPs or alleles contribute to which population?
Yeah, so, well, you can look at the length p vectors that define the principal components and ask sort of what weighting is being used on each SNP to define these positions. And some SNPs will be weighted more than others. Ideally, what you don't have is that they all weight on one or a few locations on the genome, because then what you're seeing is not genome-wide structure that might be relevant for understanding population history, but just the history of one or a few regions of the genome. Yeah, so that's relevant there. You can even zoom in within Switzerland, where we had information on French-speaking Swiss, German-speaking Swiss, and Italian-speaking Swiss, and see that they sort of shift towards the France, Germany, and Italy samples. Okay, so with these large data sets, you can detect structure that's relevant for understanding population history, how important geography has been in history, even though the structure is very, very weak. So 0.3% of the variation on this first axis. And that's actually related to the FST from European data is about the same scale. Okay. Okay, so some comments here. The number of latent factors to consider is a key challenge in each case. So when you're doing structure admixture, it's how many source populations do you fit, right? So I said there are K populations you're going to fit these individuals as having their admixture into, but how do you choose that K? In PCA, the number of components to plot. There's a result that if you have K discrete populations, you should have K minus 1 significant principal component values. But sometimes structure is more continuous, and you can have lots of principal components that show kind of non-negligible eigenvalues, and what does, how do you interpret those lower level ones, okay? In both cases, there are procedures that exist to help choose a sensible value of K. But the general rule is that caution is warranted with those approaches. It's a kind of model selection type problem and so in practice, it's generally encouraged to do exploratory analysis to evaluate the impact of K. Okay. This factor analysis interpretation I've stressed today is suggests possible alternative approaches. So doing sparse factor analysis, as was done here in a paper that Eran, Eliezer, and I worked on with one of our students here. We looked at a model with two latent factors that were like corresponding to geographic distance, and you can think of it as a factor model with a logistic link, even though we were sort of thinking about it more straight from a geographic perspective. And then there's also been developments like logistic factor analysis, non-negative matrix factorization. So a whole bunch of approaches to look at population structure that are all kind of, you can think about in the same framework. Okay. Then, yeah. No, it's not. It's yeah. No, it's. Let's take it offline. I don't want to butcher that. Okay, so a few interesting behaviors of PCA. One is that it has this threshold effect, and this may occur for admixture as well. So it's worth understanding. This has been studied in a paper by Nick Patterson and David Reich, where they show as you increase the level of FST, you get above a certain threshold, and then it's detectable. These are p-values for tests of whether you see structure, and then it becomes apparent. And this threshold is determined by the product of the number of markers and the number of sample size. So in their notation, sample size is m, n is the polymorphic loci. So as they wrote it, it's nm is greater than 1 over fst squared. So this is interesting. It means that you might not see structure because you just have a small sample size, and then once you cross it, it becomes more clear. And this is a version of that from a paper by Gil McVean, where as you increase the number of independent SNPs, you go from cases where the blue and the red are indistinguishable, you cross the threshold, you start to see that they begin to be distinguished, and then eventually be totally clear. Okay. So, you know, in settings where, yeah. Yeah, so right. So then the math for this that's cited from Nick Patterson is based on these results on phase transitions that exist for the largest eigenvalue of non-null complex sample covariance matrices. So there's results from random matrix theory that are now being useful for understanding some of this formation of structure. Okay. Another important behavior is that if you have long areas of high LD SNPs, okay, then the principal components can just focus on explaining those. So on the European data on PC3, if you don't do any filtering to catch this, you will get huge loadings on just particular regions of the genome. These turn out to be regions of the genome where there are known polymorphic inversions. So large megabase scale inversions that lock up a large number of SNPs together. And so the principal component is not explaining some genome-wide pattern of covariance, but instead capturing that there's sets of individuals that have one version of the inversion, others that have a different inversion genotype. And so in practice, what I do is I first filter on pairwise R² of 0.8 in small sets of windows across the genome, then look for these kinds of features, and if I see them, then I'll block out that region of the genome, because for my purposes, I'm not interested in, you know, what this particular region of the genome's history is. I want the whole thing. And so when you do that afterwards, you'll get loadings that more evenly spread across the whole genome, with some areas having more influence than others, but you're capturing a signal that's genome-wide. Okay. I've just about used up my time, so I'm going to need to kind of just skip it along here. But a good question is what happens, you know, when you have discrete clusters like this, two source populations admix, it makes nice clusters. What happens when you have spatially structured data? Okay, and funny things happen. It's a good general question, right? Because PCA is a very generally applied method. Spatial structure is a very typical structure in data, even outside of genomics, right? Even temporal analysis data is like one-dimensional structure, right? Spatial structure. So it's worth learning what the answer to this question is. It turns out to be like if we have one-dimensional structure, so this could be a time series, but in my case I simulated individuals on like a stepping stone and ran PCA. PC1 versus PC2, I get this horseshoe shape. 2 versus 3, I get this pretzel shape, or sorry, fish shape. 3 versus 4, I get what looks like a pretzel. Okay, totally confusing at first. And why should PCA do this? You know, does this mean that somehow the way that this line crosses, that these individuals are very genetically similar to these individuals? You know, you got to be careful how you interpret these. These patterns actually appear in old work by Jules Lissajous looking at tuning forks shining light onto walls, and there's the horseshoe, the fish, the pretzel. These are sinusoid functions plotted one against the other. So as a function of space, PC1, 2, and 3 are just nice sinusoidal functions, and you plot one against the other, you get these patterns. Okay, so this has to do with how the covariance matrix, when you have spatially structured data, has a very unique structure that where if we arrange the individuals by their position along in this one-dimensional space, then there's a lot of mass along the just off the diagonal. And these can be conceptualized as Toeplitz matrices, which in the limit look like circulant matrices and have eigenvectors. Circulant matrices have eigenvectors that form a Fourier basis. In these Toeplitz matrices with this structure, they'll have eigenvectors that are the columns of the discrete cosine transform matrix. So because PCA's eigenvectors of these structured matrices, they have a particular structure and this sinusoidal pattern. It's actually sensible in that the first PCs are like long wave sinusoidals, and as you go lower and lower, they're finer and finer scale. It's kind of like Fourier approximations where you're capturing the big scale structure and then finer and finer scale structure. Okay, so I think I'll leave you with that thought. It introduces a lot of, or maybe this thought, that aspect has introduced a lot of confusion, and that people have looked at gradients of principal components values on geographic maps and interpreted them as expansions of people moving across landscapes. And the two-dimensional version of what I just showed you is that if we take stepping stone models and look at PC1, PC2 maps, these are 8 independent replicates, you always get perpendicular gradients in each replicate, just like you do here in this European data. And this data has no expansion waves, just spatial structure. Okay, yeah, one has to be very careful. I'll see PCA analyses of like gene expression time series that will show the horseshoe effect, and then people say, oh, there's some kind of like developmental phase shift where it changes direction. But for just a temporally stationary process, PC1 versus 2 will have that curve. So, okay, so I'll leave you with that thought and take any questions. Yeah. Yeah, great question. Yeah, so yeah, one of the open challenges in this area is how to leverage haplotype patterns more effectively. And right now what most people do is sort of filter out to avoid SNPs and LD, to avoid long ranges of LD, so that you're not capturing patterns that are driven by particular features. Like, so for instance, in European data, there's the lactase persistence mutation that has increased in frequency and taken with it a large sort of megabase scale chunk of other variants. And you could worry that lactase persistence allele, its frequency is north-south in Europe, and that the PC1 will just focus on that as a result. So by filtering out SNPs and LD, you hopefully diminish the influence of these long LD regions of the genome, although I showed you some cases where it can still pop up. Okay, yeah, so yeah, the default is to just throw it out, but other ways to try to deal with it are still being developed. The two main, approaches, and you know this, I guess, but are trying to come up with ordination techniques that are based not on the covariance matrix, but on some other pairwise similarity matrix that is based on some measure of haplotype similarity. So one of these is an approach called fine structure, where you look at how similar is each individual's haplotypes to every other individual's, in terms of these haplotype copying paths of who copies more off of who in a haplotype copying model. And another approach is to build a matrix off of IBD segments. So you ask how much, what number of tracks are shared between two individuals, or what total length of DNA is shared between two individuals, and then do eigenvectors on those matrices. So those are the two main paths I see open right now for dealing with haplotype data. Okay.
So we published a paper in medicine from Dan, and what you do is you take the correlation matrix of the SNPs that are close to each other, and you basically just multiply by, you call it sigma to the minus half, then you basically get rid of the correlation, the local correlation.
Local correlations, okay.
And that's actually, that should be optimal. There shouldn't be any other way of doing this if you know the local correlation. You typically know it by looking at either, let's say, half map data, or you can do it using the
Okay, yeah, that sounds interesting. We'll have to look at that. Yeah, yeah, yeah.
In essence, it's a similar, at some level of abstraction, it's similar to the issue of spatial evaluation, right? If you think of here you're coping with an association among the SNPs that may create confounding that you're not, that you don't want. And in the spatial dependencies, you're dealing with an association among the individuals, the observations.
Yeah.
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 Creates a confounding that you don't want.
Yeah.
So similarly, there are ways of correcting for associations among the rows. So essentially changing the inner product in which you're computing.
Yeah.
On one side, the units, or the other, the variables.
Yeah.
Right?
Yeah.
In spatial statistics is a big field. What people not fight about, but the problem with that is that if you correct for the spatial dependencies, you're likely going to wash out the weights because your signal and the spatial dependence are compounded. But there are developments that people are pursuing to try to separate things, avoid spatial compounding. Accounting for space, but avoiding spatial compounding.
And it's actually to contrast two different kind of issues. One is sort of controlling for this LD structure, and other sort of taking advantage of it. So the IBD-based approaches and the haplotype sharing-based approach, what the signal they're trying to pull out, and this is related to like rare variants. Rare variants would also have this, is the what is due to recent shared ancestry. So long segments of copying are due to recent common ancestry between populations and between individuals, and should reflect recent population structure, and may be more sensitive than using kind of LD pruned or LD cleaned data, which captures longer range levels of similarity from deeper history. So we should probably wrap up, but I'm happy to take more questions. Okay.